Here's a photograph of Mendel, and Mendel is right here in this photograph. He was a monk, an Augustinian monk. He lived in Austria back in the 1800s.
And he really liked people.
And so we will talk about Mendel because he had a extraordinarily important role in understanding heredity. So we defined this term on Monday, I believe it was, or Friday last week, recently. So what does heredity mean? What is the definition of heredity? People, what is heredity, actually? So genes that are passed on from parents to offspring, right? So exactly, transmission of traits from one generation to the next. And we have known about heredity, probably I would think that our species has known about heredity for centuries, or maybe even millennia. We've known that offspring, children, tend to look like their parents. And even this understanding heredity was of seminal importance for artificial selection, for domesticating plants and animals, right? So we started domesticating plants and animals about 10,000 years ago. That's when we started farming, started specifically propagating, taking seeds and animals, dogs and cows and goats and sheep, and corn and maize, corn and rice and wheat and sorghum and many different species, and specifically breeding them and breeding dogs, for example. If you want to make a good hunting dog, you would take parents, a female and a male, that were good hunting dogs, that were good pointers, or that could follow and find the bird. You would take the best ones and have those individuals breed, because there was some understanding that those individuals would pass those traits on to the next generation. So I think that we as a species have known about heredity for a very long time. We didn't know what it was. We didn't know what was being passed down from one generation to the next. And the very first person to study this systematically and try to understand this process of why offspring tend to look like their parents, first person to do this was Gregor Mendel. And so again, he was doing most of his work in the 1860s, and first person to analyze the patterns of inheritance. And the species that he was working with was pea plants, specifically. So he was a botanist interested in pea plants. And the whole field of our understanding of inheritance and how inheritance works, which really is the basis of genetics, is named after him. We call this Mendelian genetics, understanding how inheritance works in diploid organisms like ourselves. So let's talk about the traits, some of the traits that he studied in pea plants. So he studied 7 different traits in pea plants, and for each of these traits, there was variation. Some individuals had one trait, and some individuals had an alternate trait. So at flower color, for example, this very first one, some individual pea plants of this species produced purple flowers, and other individuals in the population of the same species produced white flowers. So you can see he could use this variation to track inheritance. If we take purple parents and white parents and mix this up, then what kind of offspring do they make? He did this with seed color. Some pea seeds are green, some are yellow. Pea seed shape. Some are smooth, some are wrinkled. Pod shape. Some are inflated, some have this ribbed or wrinkled shape. Pod color, the position of the flowers. There was this variation. Each of these traits, there were 2 variants that were present in the population. All right? These were the 7 traits that he studied. And let's go through some of the basis of his experiments. And I suspect many of you guys are familiar with these experiments. So and we'll use flower color as an example. So he started out with a pea generation. What does P stand for in this case? Uppercase P. It's the parental generation, okay? So the first generation of his experiment, he took a true breeding purple plant and a true breeding white plant. What do you think true breeding means?
Like they've made sure that it has no actual like recessive of a different kind. Like it has only dominance of purple, like no, like there wouldn't be one of purple, one of white in the genes for one of them.
Right,
They-
so you're using, so your description is exactly correct. You're using some of our current lingo, and what I'm trying to get you into is thinking about when Mendel did these experiments. So he didn't have any of these when he first started these experiments. He didn't have the words dominant and recessive, and he didn't know what the genes were. But you're right in terms of a true breeding purple plant. He would breed purple plants. He would take plants have sexes just like humans. There's males and females. So he would take a male plant that produces purple flowers, and he would mate that with a female plant that produces purple flowers, and he would breed them together and collect their offspring, and those purple flowers, those purple offspring, he would take those and take the purple male, purple female, and breed them until eventually there was never, you would take a purple, and they would only ever produce purple offspring. So that's true breeding. He basically weeded out the other trait from that lineage. Whatever that trait was, he didn't know what was being inherited. He didn't know anything about DNA. He hadn't discovered DNA yet when he was working. Same thing with the white flowers. So true breeding. So we know that he purged those alternate genes from its genome. So he bred them, took a male purple flower, bred it to a female white flower, and what he found in the F1 generation was that all of the offspring were purple. Okay? So that was the first thing that he found. He did this experiment hundreds of times, actually more than hundreds of times. He probably did this at least 1,000 times doing these breedings. We'll take a look at his numbers in just a minute. And they all turned out purple. If he took a purple male and a white female, if he took a purple female and a white male, bred them, always purple flowers. Interesting, right? In this F1 generation. So what does F mean in this case? Anybody know what that stands for? This is a word that we don't use oftentimes in our normal dialogue, but it's filial. So filial just means relating to sons and daughters. Your filial relations are your sons and daughters. Okay? So the first filial generation, the first offspring generation, all purple. Then what he did is he took a male F1, so a male plant from this generation, and a female plant from this generation. They were both purple, and he crossed them to produce the F2 generation, the second filial generation. And what he found was this classic 3-to-1 ratio. He found when he counted up all of the seeds, he grew up the seeds of the F2 generation, that for every one white flower, and so the white trait reemerged, saw some white plants produced then, but for every white flowered plant, there were about 3 purple flowered plants in that generation. All right? Raise your hand. I'd be curious to see, anybody heard of this 3-to-1 ratio before, sort of this classic 3-to-1 ratio? Hopefully you guys are thinking, what's going on, what's happening here, and trying to refresh your memories about all of this. Some of you guys, I'm sure, know. We'll talk about the details of what is going on here. So, to understand what's happening, we can use a Punnett square to understand inheritance, to highlight the 4 possible offspring that can be produced. Okay? Punnett squares. You guys heard of Punnett squares before? You guys like Punnett squares? Yeah, sometimes there are moans with Punnett squares. Anybody moan when they hear Punnett squares, think of Punnett squares? You guys all like Punnett squares. All right, so Punnett squares, let's go through this. But before we do, so this is a Punnett square that we would set up. And so essentially what you're doing with the Punnett square is you're taking, what is this symbol? Does anybody know what this symbol is here?
Male.
This is the male, so this is the male plant, and these are the genes that it has. So you would put, because pea plants are diploid, so diploid, what does diploid mean?
They have 2 genes, 2 chromosomes from each parent of each site.
What is diploid? Can you speak louder? It has 2 copies of every chromosome, right? So for every gene in a diploid organism, and pea plants are diploid. Are we diploid?
Yes.
We are diploid. Every multicellular eukaryote is diploid. Every plant is diploid. Well, with some exception. Well, let's take that. There are some exceptions. Asterisk. All with an asterisk. Certainly most plants are diploid. Essentially all animals are diploid. They inherit 2 copies of every chromosome. So that means that if you have a gene for flower color, for example, and if flower color is coded on a particular chromosome, if you're diploid, you've got 2 copies of that gene because you've got 2 copies of that chromosome, right? So a Punnett square is just acknowledging that the organism is diploid and that it has 2 copies of every gene. And so for the flower color gene, we know pea plants must have two copies of the gene that code for flower color. And so the male, remember when we were just talking about meiosis? Meiosis is the having of the chromosome number, right? So producing gametes, producing sperm cells or egg cells. And sperm cells or egg cells are only going to have 1 chromosome of each pair. So a sperm cell or an egg cell is either going to have chromosome copy 1A or 1B, for example. So that's what a Punnett square is simply looking at the possible gametes, the possible sex cells that an individual organism can produce and make. That's what a Punnett square is getting down to. So, and then this is the female, so if we cross a male by a female, and if these are the genes that she has, if this trait is coded on chromosome 8, for example, she has 2 copies of chromosome 8, she has 2 copies of this gene, and this is the genotype of each of these genes. We'll talk about this in some detail in just a minute, what we mean by this. And then the Punnett square, you just fill this out. Each square of this Punnett square is a possible fertilization. Reuniting, taking a sperm cell. If this sperm cell right here fertilizes this egg cell, this is going to be what we call the genotype of that possible offspring. And so a Punnett square, there are 4 possible offspring that can be produced. All right, that's the basis of what's happening. So I've kind of described this. I've gotten ahead of myself. Pea plants have 14 pairs of chromosomes. So let's say that flower color happens to be coded on chromosome 8. And flower color is a protein that is produced. It's a pigment protein. So to make that protein, it's a piece of DNA. And I don't know if it's coded on chromosome 8, but let's just say that it's coded on chromosome 8, and it happens to be coded right here where I've made these marks. So an individual is going to inherit 2 copies of the flower color gene. Maybe this gene, one of these genes might be, the possibilities are purple or white, right? And the difference is just a different DNA sequence. Very small difference. I'll show you the difference between them in a few minutes. So an individual, again, when we talk about meiosis, there's independent assortment of chromosomes. You guys remember when we talked about that? So when the chromosomes in meiosis 1, when the chromosome pairs split and go to each pole of meiosis 1, that first round of meiosis 1, the job of meiosis 1 is to make the cells haploid, to make them have 1 copy of each pair of chromosomes. And they assort independently in every egg cell and every sperm cell. So in this example, this egg cell that was produced happened to get chromosome copy 8B. Got this copy of the flower color gene, whether that happened to be a white copy or a purple copy. An individual pea plant could inherit 2 copies of the purple gene, could be 2 copies of the purple gene, could inherit 1 copy of the purple gene, 1 copy of the white gene, or could inherit 2 copies of the white gene, right? And the offspring that it's going to produce is going to depend on that particular egg cell and sperm cell, which copy it got. And it's all based on this independent assortment of chromosomes in meiosis. Does this make sense to people? Does anybody need me to explain this differently? All right, so I'm using, of course, I'm using vocabulary that we used on Monday. You guys hopefully all learned all that vocabulary because it's much more difficult to understand what we're talking about today if you didn't sort of internalize all of these terms on Monday. So let's use a Punnett square, knowing now what we know about the inheritance of these. Let's take a look at Mendel's experiment, the P1 generation, using the P1 generation to generate the F1 generation, and then what's happening when we use the F1 generation to generate the F2 generation. We'll do this, we'll take a look at this, his experiments, filling in Punnett squares, okay? So let's say that this purple individual, in this case, the purple individual was a true breeding purple individual, had weeded out all of any white genes from that lineage, so those purple flowers inherited 2 copies of the purple gene. When we go back and take a look at this, a purple individual has a purple gene and a purple gene. Both chromosomes are purple. And in this case, we know that purple, or we will say, and I'll describe this why, purple is big P in this case. So the male, when a male true breeding purple plant produces sperm, it's only going to be able to produce sperm that has a big P allele because both of its flower color alleles are purple, right? So in our Punnett square, one possibility is that on chromosome 8A, a sperm will inherit chromosome 8A, which is the left side of the Punnett square, or chromosome 8B, which is the right side of the Punnett square. In this case, just so happens that both chromosomes are big P or purple. They're identical, okay? So far, so good? And then the female, we can look at the possible eggs that a female plant can make. Again, the female's here on the y-axis of our Punnett square. The female is true breeding white. That means that both of her genes, both of her flower color genes, are the white genes. In our nomenclature, we'll call them little p. So we've got big P is purple, little p is white. So she's only able to produce eggs that are little p, or white. And so these are the pops. So she can make a little p egg or a little p egg. Happens to be identical. So far, so good. And then we can fill in the Punnett square. Can you guys fill in this Punnett square? Mm-hmm. You probably can all do this conceptually, right? All of the offspring, what is the genotype of every single offspring going to be in this cross?
Big P, little p.
Because remember, the diploid number gets restored upon fertilization. So if the fertilization happens to be this top left one, the genotype of the offspring is going to be, whatever it is when we fill this in. And then this is another possible offspring. Each of those squares represents a possible offspring, possible restoration of the diploid number. So what is every genotype going to be? Big P, little p. Exactly. So every single offspring is genetically identical as far as flower color is concerned. One gene in the genome, and they're all big P, little p, and they all look purple. Mendel found out that they all looked purple, so this was an important thing. Now again, he didn't know anything about chromosomes. He wasn't able to look at anything like this. He, again, didn't know what DNA was. He didn't know what proteins were. All of that wasn't discovered until the 1940s, 1950s, 1960s, until we started figuring out exactly what was happening. But Mendel is just looking at traits at this point in time, right? So that they're all purple. So then, what he did to produce the F2 generation is he just took one individual, a male from this generation, who had a genotype big P, little p, and he crossed that with another purple individual from the F1 generation who had a genotype of
big P, little p.
Big P, little p, right? Because they all have big P, little p. And he crossed them, so we can make a Punnett square here as well for the F2 generation. So if you cross a male, that's big P, little p, because they're all big P, little p. Cross that with a female, here on the y-axis, big P, little p. So again, the chromosomes that these individuals have, if we come back here, chromosome 8, we look at chromosome 8, one chromosome is a purple allele, a purple copy of the flower color. The other chromosome has a white copy, right? For every one of these individuals. Can you guys fill in this Punnett square?
Mm-hmm.
Everybody can fill it in? Why doesn't everybody fill it in? What are the possible offspring that can be produced from a cross big P, little p, by little big P, little p? Anybody having a hard time doing this? I think there's real value in sort of puzzling through things and actually thinking about things. So, actually had a former student graduate. Just I'll give you this little anecdote as you guys, as the few of you guys work through this Punnett square. A former student came back and visited me a couple years ago, and he said that he wished he was in graduate school now, and he said that one thing that he wished he knew when he was an undergrad was he wished that he approached his classes when he came to class the same way that he approached going to the gym. So when you go to the gym, imagine you go to the gym and you just kind of like, you're going to go and work, lift weights or ride a bike or something. But imagine you go to the gym and you just kind of like walk around and talk to people and chill out. Is there anything that you get from that experience in terms of what your goal was in terms of physical exercise? Not really. You're kind of not taking it seriously. You're not really engaged in what you're doing. And he said, when he goes to the gym, he likes to be serious. He does the work. He's very intense, and his goal is to exercise his body. And when he wishes that when he would go to class, he thinks that the mindset should be the same. That when I'm in class, it's like a mental workout. That I'm engaged and I'm constantly firing my neurons, and I'm trying. When the professor says, make a Punnett square, do it. Make that Punnett square. Be engaged. Be physically firing those neurons. And come out of here, come out of your time in all of your classes sort of mentally exhausted. And don't be taking vacations. Don't be walking around the gym, outside of here, or daydreaming, or on your phone, or doing other things. If you take this seriously, you're actually going to get more out of it. Your time is going to be better spent. Okay? So did you fill out the Punnett square? I think you did. Did everybody do it? So what is this? What's going to be in the top left square? What is the possible genotype of the offsprings? If a big P sperm fertilizes a big P egg, that offspring is going to have what genotype? It's going to be big P, right? Actually, let's just fill in. So they're going to be big P. This individual, you just bring it over, right? So this big P comes over here, the little p comes down. It's not hard, you just fill it in that way. So these individuals are going to inherit a big P, little p, and then this individual, that's the only one. 1 in 4 offspring are going to inherit 2 little p alleles, And so this is what he found. Any individual that inherits at least one copy of the big P allele is gonna have purple flowers. So they can have 2 copies or 1 copy of the big P, they'll be purple. If they inherit 0 copies of the big P allele, they will be white. So this is what the classic 3-to-1 ratio, this is what it derives from, is this idea of what we now call dominance and recessive. We'll talk about that in a minute. Well, actually, before we do this, before we go over this, why don't we take a break, 2-minute break. You guys can pull out your phone.